I hope you can all hear me. Several things I want to do today. First, let you know that a week from today, next Wednesday, the 1st of September, I'll have quiz 2. And quiz 2 will cover everything since quiz 1, and that would include cell structure and function, life in the universe, and classification of organisms, which we'll go over today. So it'll be very similar in style to quiz 1, a mixture of question types, and worth 10 points with 15 minutes allowed. And you'll have all day on Wednesday, next Wednesday, week from today, to complete it. So there are several things I'd like to do, starting with
Professor, I have a quick question.
yes, your name?
Yusuf Faraj. I know you said that class on Monday would have ended earlier, like 15 minutes earlier, but I'm pretty sure we got out at the same time. Like, did you just forget, or does it just not actually end earlier if we have a quiz?
It should end earlier. It's my fault. I ran overtime.
Oh, okay. I was just making sure. So next week on Wednesday, the class will end at like 11:15. Or I mean, 1:15. My bad.
No, it should end at 1:30.
Oh, does class normally end at 1:45?
Yeah, so
Okay.
I subtract the 15 minutes that you use some other time during the day for your quiz.
Okay, thank you.
You remember from last time, there were some questions. Let's see. That we just acknowledged they were there and we didn't do anything about it. So what I'd like to do now is go back and review those questions. So here's the first one. Let me make this bigger. The human body contains 230 different types of cell. For example, skin cells, blood, nerve cells, and so on, 230 types. Each of those cells contains about 23,000 genes. So a set of genes, and it's the same set in each cell. So can you come up with an explanation for how this can happen?
Professor?
Yeah, your name?
I can. Alex Alos.
Okay.
You can hear me? All right.
Yes.
I guess my hypothesis on why is probably because they have like a different function for each genetic material. Since you said like skin, blood, and nerve, they probably look similar, but I'm guessing because they have a different function, they also function differently.
Yes, they certainly function differently. Anyone else have an explanation?
Yeah, I'm sorry. What exactly was the question again?
Can you come up with a hypothesis to explain why different cells, they all have the same genes, but the same cells have different functions?
Maybe because they're all making up as like a singular organism. So together they need to form, a liver, a heart cell, different functions, like Alex was saying, in order to build up one organism. And I think I read that because they're smaller cells, there's less surface area compared to one cell. So it's more efficient that way. I don't know if I'm saying that right, but
Yeah, the latter part of your statement refers to the question, why aren't we just one big cell?
oh.
But getting
uh,
back to the point, yeah, you've come up with an observation, but can anyone think of a mechanism for how?
I have an idea, maybe. Yolanda Dukes.
Yolanda Dukes, okay.
And I was thinking, well, maybe because you have two different people that are actually donating to the cells, two different, like the mother, father, whatever genes that are usually hereditary. That maybe that's why they're different, but yet they have the same function, like chromosomes.
Well, this
I'm trying to say
question really doesn't have anything to do with heredity.
the cells. Then okay.
So anyone else have a thought about it?
This is Gabriela Galvez.
Okay.
I know that cells contain proteins, so maybe because they have different kind of proteins. I know.
Well, yes, you're right. Different cells have different kinds of proteins, but why?
Professor, it's Alicia Carroll.
Okay.
Is it because each cell is grouped and they form tissues, and those tissues perform different things like muscle tissue, and then the tissue turns into systems, and then we have like circulatory systems?
Yeah. Again, you're describing what we observe, but so far no one has told me
Professor,
why.
this is Leonardo.
Leonardo, your last name?
Cuellar, CUE.
Oh, okay, Leonardo. All right.
I'm thinking maybe some sort of external factor changes how the DNA is read. Maybe like parts turn off, parts turn on.
Aha, you're on the right track.
Professor,
Okay.
can I take a guess?
Yeah, your name?
Elizabeth Ortiz Martinez.
Okay.
The question is saying that they have the same genetic material, but they look and function differently. So does that mean that it's because of the environment that they're around, like in our different parts of our body, even though they're the same cell?
Yeah, you're talking about a response to stimuli. And so let me just get to the point here. Appreciate all your thoughts and your comments. The answer in a word is regulation. So you have 23,000 genes, but if you take any given cell at any point in development, only a small number of those genes are actually operating. They're actually making proteins. So if you look at, say, a skin cell, all the proteins that are needed to make skin, the genes for those proteins would all be turned on. The genes for blood, nerve, everything else, in other words, most of the genes in the cell, in any given cell, would be turned off. So whether a gene is on or off, as several of you have said, is in response to stimuli, either internal or external stimuli. So we're going to talk a lot more about how genes are regulated. There are lots of different mechanisms by which a gene is either turned on or turned off, or the speed at which it's running is regulated.
I have a quick question. When you
Anthony, Emilio?
Yeah, so when
Okay.
you say that they can be expressed based on external and internal. Internal, would that also be considered like your emotional states and things of that nature?
Yeah, it could be a response to emotional states because emotional states
Or
reflect
stress?
hormone levels, and hormones are certainly stimuli.
Got you. Okay, cool. Thank you.
Okay, I want to move on to some other questions. All right, so here are some example problems, and this gets back to telescopes and what you can see. Not telescopes, but microscopes and what you can see. So here you are looking at a cell under a compound light microscope. It's approximately 50 micrometers in diameter, contains a visible nucleus and a cell wall. The question is, what type of cell is this? And into which domain of life would it be classified?
Sean Coke here.
Okay,
It's a plant. If it has a cell wall, it's a plant.
okay, well, prokaryotes also have.
It's an onion cell that's viewed with a light microscope and is the domain of Eukarya. My name is Wesson Raffo.
Okay.
And as my classmate said, it's a plant cell.
It's a plant cell, I think you said because it has a cell wall.
Yeah.
But prokaryotes are not plants.
No,
and
eukaryotes.
They also have cell walls.
I said, yeah, no, it's for the eukaryotes. Prokarya.
Okay, it's a prokaryote, and it has a cell wall. And it has a visible nucleus. So yes, because it has a cell wall and it's large, it's probably a plant cell. And we won't talk about how it's classified because that's the topic we're going to talk about later today. All right, let's go to the next one. You're looking at a cell under a microscope that is broad shaped and about 4 micrometers in length. Does not appear to have any internal structures, but it does have a cell wall surrounding the cell. So what type of microscope are you looking through? Remember the different types. We have a light microscope and 2 types of electron microscope.
This is Wesson Raffo. I think it's a scanning electron microscope.
A scanning electron microscope. And why do you say that?
Because it's like just outside, not, it's just like seeing the internal and the external.
Right. It doesn't appear to
Yeah.
have any internal structures,
From
but
the
it does
outside.
have a cell wall.
Mhm.
And so this would fit with a scanning electron microscope. Is there another possibility?
Excuse me, this is Trinity Daniels.
Daniels, okay.
I would think it's a light microscope.
It could be that. So that's another possibility here. It could be a light microscope at low magnification. So you can still see it, but the magnification is too low to see the internal structures, but you could still see the external cell wall. So it seems to me that this one has 2 possible answers. It could be either a scanning EM or a light microscope. And finally, this one, you're viewing a structure that's less than 0.02 micrometers in length. Does not show any type of internal structure, no organelles. What type of microscope and what is it? no internal structures.
This is Steve Stinson, and I think it's got to be a scan electron microscope.
Scanning electron microscope. Yes, that would be consistent with the fact you can't see anything internally. So look at the size. It's 0.02 micrometers, which is quite small.
Very small.
You can't see internal structures. So it could be a scanning electron microscope. You could certainly see that with it. Could it be a light microscope?
I don't think so, but I could be wrong.
No, I think you're probably right. This is too small to be seen even with the most powerful light microscope. So it's probably a scanning electron microscope. And it's probably a virus. All right, that's what I wanted to do to finish up with that slide set. And next on the agenda, yeah, we have a worksheet that's due today. So I want to go over the answers to the worksheet. You see the worksheet? Methods of scientific inquiry?
Yes.
Okay.
Yes.
Okay, the first question is circle the letter of each question that can be answered through science. I had said indicate in whatever way you wish. Well, a number of you misinterpreted this, and this gives me an opportunity to point out that always read the instructions to a question very carefully. So quite a few of you actually answered the questions, and you said 212 degrees Fahrenheit. What temperature does water boil? Well, that's not what the question is asking. It's asking, can that question be answered through science or not? So I've highlighted here in yellow all those that can, and in fact, only 2 of them cannot be answered through science. B, how can my team work better? And G, is it hot or cold today? Those are not scientifically based questions. The next one is indicate those statements that are true about graphs. The first 2 statements are true. The graph can reveal a trend. Graphs help scientists interpret data. The last one is not true. The next is, which of the following are theories, which are hypotheses? If hormone Q is applied to the leaves of the plant will grow faster. Well, that is a hypothesis. It's not based on substantial evidence. Well, the next one, plants grow best in the absence of light. And I've indicated it depends on how you look at it. You could interpret that as being either a hypothesis or a theory. It's well established. I think no one has ever seen a green plant grow in the absence of light. So through many, many observations over long periods of time, I think it's well established that light is beneficial to plants. So if you put a theory, I accepted that, or if you said, oh, it's only a hypothesis, I would accept that too. All living organisms are composed of one or more cells. That is the what theory? The cell theory, which I told you last time. Make sure you know the 3 elements of the cell theory. Number 4, some human diseases are caused by microorganisms. And the key word here is some human diseases. And that is well established. It goes back to prehistory. That observation has been made. So that would be a theory. Matter is composed of discrete units called atoms. It's well established. That's a theory. And the last 3 are all hypotheses. Then we have some two thought questions. First is about Bob wants to grow lots of pink flowers, and he wants to test fertilizer. And what's wrong with his experiment? Well, most of you got this. He didn't put the plants in the same environment. Some he put on the windowsill, and some he put, where was it, in a closet or on a shelf. So he should have had all the plants in the same location, the same amount of water, the same amount of sunlight, and varying only the fertilizer. So most of you got that or something resembling that correct. And the next one is about Jenny and her super green brain snacks. What's wrong with her experiment? Well, there are a lot of things wrong with it. First, there are no controls. There are no girls that are not eating snacks at all. So there's no control group. There's no plan for how these data are going to be analyzed. If you look at the data, they don't indicate any trends. Even without graphing them, they don't indicate any trends. Some are up, some are down, some are essentially no different. So this is a case where you really need statistical analysis to see if the snacks are working. And the final thing that a number of you pointed out is that you can't, in an experiment like this with humans, you can't really control everything. So what is the diet? Are all the girls eating exactly the same thing at exactly the same time every day? Do they all sleep exactly the same number of hours a night? Do they go to bed at the same time, get up at the same time? Do they all have the same study habits? So this is something that Jenny made no attempt to control. And as I said, it's very difficult to do this kind of control. And the last point here is that this is a good time for you to think about why it's so difficult to do clinical trials. Because clinical trials have to be done with humans, and yet it's impossible to have a cohort of humans that are all the same genetically, that all have the same lifestyle, and so on. So clinical trials can be done, but with great difficulty and with great expense. Okay, so most of you have done this worksheet. I've graded most of them, but a few are still left to grade. Are there any questions about the worksheet before I go on? Okay. Can you see the quiz? Oh, I don't think you can.
Yes.
No,
No,
No.
No.
Now can you?
Yes.
Yep.
yes.
All right, good. All right, this is a quiz that I've graded all of them so far. I'd like to go over the answers. First one, true or false, biology is one of the natural sciences. It's a branch of science that deals with the physical phenomena on the planet Earth. A lot of you didn't get this. And again, I want to emphasize that how important it is that you read the question carefully. So here, it's a branch of science that deals with physical phenomena, and yet we're talking about biology. So the science of biology deals with life, and physical phenomena are not alive. So if we were talking about physical sciences, we'd be talking about things like geology or soil science or chemistry, physics, and so on. So if this had said with physical and biological, or biological instead of physical, then it would have been true. But as it's stated, it's false. Number 2, which of the following is not a property of life? And this refers back to your textbook, where on page 6, there were 9 or 8 different criteria which reflect a property of life. And this asks for those items here that are not properties of life. So reproduction is, response to stimuli is, heat absorption, no. A rock can absorb heat. Growth and adaptation are life types. Combustibility, no. Regulation, yes. Homeopathy, not sure even what that is. And transmissibility are not properties of life. So these 4 items that are highlighted are the correct answers for that one. On number 3, a lot of you had problems with this. And again, I want to come back and emphasize, read the question carefully. So it says, this is matching, match the best fit partner. So it could be that there's more than one correct answer, but I'm asking for the best fit. And so the best way to approach a problem like this is by the process of elimination. So look at one that you know the answer to. So single-celled is pretty easy. That's number 1, simplest form of life. Toad is not so easy. Energy processing is not so easy. Polar bear is clearly, of all the choices, it's adapted to extreme cold. So polar bear is number 2. California redwood tree, you look here and you can see that it could be several of these, but it's huge. So it's easy to put down. That's a case of extreme growth. All right, go back to one that you haven't done yet, energy processing. Well, what's left? Locomotion is left. Highly organized set of tissues and organs is left. Either of those could be right, but you need number 4, highly organized sets, tissues and organs for something else. So what's left is locomotion. So that's number 3. And the only one you haven't done yet is toad. And if you look at what's left for toad, it's number 4. It's the only one left that fits. So is there any question about, first of all, any of these answers, but secondly, my main point here is how you approach a question like this, and for things that you're unsure of, leave them for the last and approach it as a process of elimination.
Sean here. I have a question.
Sean?
So just to be clear, energy processing and regulation are two different things. Adaptation to extreme cold is what, regulation then?
Adaptation to extreme cold is a polar bear.
No, I understand that. I'm just saying, just to further understand, the adaptation to extreme cold is also known as what?
I guess you could say as a response to stimuli, the cold being the stimulus. So over a long period of time, polar bears have learned to adapt to extreme cold.
That's a form of internal regulation, or?
Adaptation to extreme cold. Well, certainly there are regulatory mechanisms that allow a polar bear to live in extreme cold. So yeah, you could view this as a regulatory phenomenon.
Okay, thank you.
Okay, I think everyone got the next one right. Smallest fundamental unit of matter is the atom. Actually, a few of you didn't get it right. You put cell. That would have been correct if it had said the smallest fundamental unit of life would have been the cell. This is a unit of matter. And number 5, write a short statement giving an example of how knowledge of biology impacts your life and what would happen if you didn't have that knowledge. I had a huge wide array of different responses to that. I gave you all full credit for saying what you thought, unless you said something that's totally wrong. And most of you did not do that. Okay, from the list below, indicate features that are common to both theory and hypothesis. And these, somebody's got the mic on. Could you turn it off, please? The two similarities are falsifiable and testable. And I've got here the table that we went over in class showing similarities, hypothesis and theory are both testable and falsifiable, and they both have a common goal. One of the possibilities here was a suggestion. Well, a suggestion is only for hypothesis. Based on multiple tests is only for theory. Specific to an observation is only hypothesis. And starts with a conclusion is actually neither of them. So the two answers are falsifiable and testable. A controlled experiment is one in which everything is held constant except for one variable. That's true. And I've included here, there are several definitions of a controlled experiment. This is just one of them that I chose. All variables in an experimental group and a comparison control group are kept the same except for one variable in the experimental group. So this definition supports this statement in the question. And finally, on the following graph, indicate what is the dependent variable. The dependent variable is the y-axis, and the independent variable is the x-axis. And some of you said time and velocity. That was fine. Okay, any more questions about the answers to the quiz?
Professor,
Yes.
this is Fakriya. I just want I have two questions. So the first one is there's some vocabularies that the biology vocabularies, and sometimes me and I don't understand the vocabularies, or if just in case if you put like definition, because we since we are running out of time for the quiz, and make me like more like the pressure like on the vocabularies, and I didn't get that. So yeah.
Okay, I don't understand your question.
Oh, I'm saying like the question is like there are some words, the vocabularies, the biology vocabularies that I don't understand. And since the time is less, and like I don't understand the meaning. And so if you put like some definition, like what does that mean, so we understand the better way.
Okay, so you're saying that you didn't have enough time to understand what the question was asking?
Yeah. And also the definition, the word.
Well, I don't ask for any definitions here in any of these
No, like
questions.
there's some words like in a sentence, like there's some words like I don't understand like the meaning of that.
I see. Okay, if you have difficulty reading and understanding words, you should consult with DSPS and
Okay.
Get a form from them and have it forwarded to me, and then I can make a correction for you.
Okay. All right, thank you. And the second question is like, can you please increase the timing, like because there's some written question and like matching, and it like it's a lot of pressure, like we are running out of time, and if you increase like the timing for the quiz.
The average
I agree
time
with that. I
taken.
run out of time as well.
Okay, the average time taken for this quiz was 13 minutes out of 15.
Oh,
So
15 minutes. It's only 15 minutes. And please, I could ask you the same question. Could you please incur more time for myself? I need more time. 50 minutes is much.
Okay,
I was so stressed. And I hope you can give us more time.
You understand why the time is limited. I explained that in the syllabus. That is because these are closed book tests, quizzes. And if I gave unlimited time or extended time, it would give plenty of time for you to use other resources other than your brain to answer the question. So that's why I limit the time. So if you have a problem with time, consult with the SPS. And if you have a good reason why you need more time, I can grant you specifically more time. But first I need evidence from DSPS.
Well, I have a question. This is Steve Stenson. I'm hearing them with the time, and I know time ran out for me. I just happened to get to the last question and get it in, but time ran out, was running out because of typing. You had, I guess, a couple short answers that you wanted. And I know that you probably have lots of students because you said 13 minutes was the average time. But I'm thinking that if you gave 5 minutes more, that might be enough for that. If you're going to have written questions that you that people have to answer and they have to actually type stuff out.
There will almost always be essay questions. And my comment about that is time management. So if you see an essay question that's asking something that you know you're going to have to give some thought to, save that for last. Don't sacrifice the easy questions trying to do a hard one.
Can I just make a comment about this?
Name is?
Oh, Elizabeth.
Elizabeth.
Well, if some of you are not aware, the time limit is usually set by, well, it is set by the College Board, and you're supposed to take about a minute in each question. That's why there was even a little too much time in this quiz.
Okay, you're saying that there is a code indicating how much
I
time.
mean, I took AP classes in high school, and they always told us that the College Board would put on timings because you're supposed to take around 1 minute in each question if you actually know it.
Yeah, well, you bring up a good point, and that is to prepare, you should prepare for a quiz or a test. So I expect you to know the material in advance so that when you see a question, you know the answer. I don't expect you to go to the quiz and read a question and then have to sit and ponder, do I know the answer to this or not? If you study well in advance, you should know the answers to the questions. And as you just said, Elizabeth, 1 minute per question for most of the questions is actually more than you should need.
Professor, I understand the lady point, but in my case, I'm a ESL student, and it's sometimes it's really hard for me because it's not my language, and I try to do my best. But
Okay, you're a good, you're a good candidate for DSPS.
Yeah, but in my opinion, I just want to ask you, for 50 minutes, maybe 20 or 25 minutes, it will be fine. But I understand that. So thank you so much.
Okay. Anything else? Anything? Yeah, someone?
Yeah, Yolanda Dukes.
Huh?
I was on the oh, I can't push it up. Oh no. Well, anyway, yeah, I get what the guy was saying, but like I said, I didn't get through it because I was just kind of viewing the quiz, and then I found out I was actually taking the quiz and I didn't know, so I ran out of time. But I was thinking you had the 100 words for the essay questions. Yeah, and I did try to skip that and run on to the next one. But like I said, I was viewing the quiz as I thought I was and not actually taking the test, which it actually made me take the test, and I ran out of time, so whatever.
Well, the other point to be made here is that this is the first quiz. Okay. And you're getting to know me, I'm getting to know you. Yeah. And so you'll have a better idea next Wednesday for quiz 2, what to expect.
Okay. Okay, so don't be trying to view it no more. Actually go on there and know you're gonna take the test. Okay, I got it.
Okay. I should get your pencil and write this down. I said that the average time for this test was 13 minutes. The shortest time, and several people had 9 minutes to complete this test. So clearly, you're a highly diverse group, and you do things in different amounts of time. But the average score for the test was 74%. The highest score was 94%. Life in the universe. I mentioned this a couple of times previously when we were talking about the sizes of living things. It raised the question, is there life elsewhere in the universe beyond Earth. This is a biology class, so life is of interest to us no matter where it is. I remind you, you've seen this slide before, remind you that matter and living things range from very, very small to intermediate to very large. And for the very small things, we can't see them with our eyes, but we can see them with various types of microscopes. And so going beyond what we can see with our eyes are very large things. And for very large things that are beyond our own planet, we need telescopes. And I mentioned there are telescopes with lenses, there are radio telescopes, there are space telescopes, and this is an image through one of the space telescopes. It captures in this one very small section of the sky, 15,000 galaxies. And the last time I talked about what a single galaxy is, our own galaxy, the Milky Way. It's 200,000 light years across, and I discussed what a light year is and how 200,000 of them would be 5.8 trillion miles. And within the Milky Way galaxy alone, there are approximately 300 billion stars, and probably most of those stars have planets. So this is a space telescope, a particular one called the Kepler Space Telescope. It was launched in 2014 and it was retired in 2018. The goal of it was to go out into space without the interference of Earth's atmosphere and look for planets. And you can see it, there are many solar panels on it. That's how it received its power. And so the Kepler Space Telescope discovered a lot of things. And so among the billions and billions of stars, if you remember Carl Sagan, the astronomer, billions and billions of stars. Most stars are likely to have planets. And so there are even more billions and billions of planets than there are billions of stars. So because of these astronomical numbers, what's the probability of life on any one of them besides our planet? What are the conditions that are required for life? Well, exobiologists assume that liquid water is necessary, that life, at least as we understand it, can't exist without liquid water. And to get life started, you need an atmosphere that has nitrogen and carbon dioxide in it. And the size of the planet is critical because of the gravitational field. It can't be either too large or too small. So this bears on the issue of the state of water, liquid or solid or gaseous. So that this thermometer here shows when water freezes at 32 Fahrenheit, it boils at 212 Fahrenheit, and you can see that the Earth here falls in that range. And some of the Kepler planets are also close to that. Jupiter and Neptune are way too cold for life. Mercury, Venus are way too hot. Most of the Keplers are way too hot, and a number of them are way too big. their gravitational fields would be too big. So the point here is that if one of your criteria is that water must be liquid, a lot of planets are eliminated by that criterion alone. Okay, there are what are called the Goldilocks conditions for life. You remember the story of Goldilocks? Not too hot, not too cold, not too big, not too small. Had to be just right for her to use it. So that applies to planets as well. They can't be too hot or too big or too cold or too small. A planet has to be just right. Well, one of the things that the Kepler Space Telescope found was a planet called 186F. It's the most Earth-like planet found to date. And here you see a size comparison between Earth and Kepler 186. You can see our solar system and the Kepler 186 system. In both cases, you see the star. Here's our sun. Here's comparable star, roughly the same size. Kepler 186 has several planets, B, C, D, and E, and F is way out here in this orbit, but that makes it about the right distance from its star to have the right temperature. And you can see it's about the same size as Earth, so it has about the right gravitational field. So here are the criteria by which Kepler looks like it's amenable to life. It probably has liquid water, or could have. It probably has an atmosphere. Don't know about its magnetic field, its liquid core, but it does have the right size, the right mass, to not be either too big, in which everything would be crushed to the surface, or too small, in which case the atmosphere escapes. So these Goldilocks constraints greatly reduce the number of prospective planets. So even though there's this enormous number of stars, enormous number of planets, the ones that could conceivably have life as we know it are, when I say relatively small, it's still in the billions. Okay, let's think about what are the chances of we humans encountering life somewhere else. Well, one big issue is distance. So just for the sake of relative distances, consider these. From the Earth to the Moon is about 1.29 light seconds. So it takes about a second and a half for light to reach us when it's reflected off the moon. From Earth to our sun is about 8 minutes. Earth to Mars, 12. To the nearest star is 4.3 light years. Remember what a light year is. It's the distance light travels in a year. And from here to the other side of our own galaxy is 52,000 light years. Well, this is an enormous distance. We can't even imagine 52,000 light years, how far that is. So if we have to go somewhere to find life, it's very unlikely that we'd ever be able to do it. So if you just think about Kepler 186F, the most Earth-like planet that is known, it's 500 light years from Earth. So 500 light years is 2.5 times 10 to the 15th miles. It's a huge distance. Kepler 186F is in our own Milky Way galaxy, which doesn't help much, the time to get there from Earth would be 500 years at the speed of light. Well, at this time, we can't travel at the speed of light. Who knows if we ever will be able to. But at the speed of a rocket, a fast rocket escaping from the Earth, it would take 242,000 years to get there, to get from Earth to Kepler 186f. So it's not feasible for us to even think about traveling to Kepler 186f and see if it has life. So distance is a huge impediment. The other factor that we have to consider is what form of life are we expecting to find? We assume, since the only example of life that we have is here on this planet. We don't know if in other places life would have the same form or very different, but for purposes of considering it, we have to say, well, we have to assume that life elsewhere has the same basis as life here. So on planet Earth, the prebiotic ocean contained about 10 to the 24th liters of water, was rich in amino acids, about 1 micromolar concentration. When peptides got to be 32 amino acids long or more, they were able to self-replicate. And once they did that, with the Earth being 4.5 billion years old, 10 to the 40th self-replicating peptides per billion years. So that's an enormous number and many, many opportunities for more complicated forms to become the type of life that we see around us. So life on Earth can be traced back 4 billion years. Most of that time, life forms were very primitive. So we still have evidence for some of them, like fossilized trilobites, bacterial communities in thermal vents, which are very hot. They're about the temperature of boiling water. So the point is that these are not intelligent forms of life. They're not even highly complex. And only in the last few hundreds of thousands of years have we had complicated forms of life on this planet. So about 0.0001% of the history of life on the planet have we had anything close to an intelligence. So the conclusion from this is that any life forms on other planets are likely to be primitive, very unlikely to be intelligent. And this helps you visualize that problem a bit better. We have a time scale here in billions of years, from 5 billion to the present. So 4.5 billion years ago, our planet formed. And about 1 billion years after that, life on this planet formed, single-celled life. And then a little more than 1 billion years after that, we started seeing multiple multicellular forms, which got more complicated. And then about 600 million years ago, we started seeing vertebrate forms of life, like fish. And then about 360 million years ago, life forms started occupying land, came out of the water. And it's only about 7 million years ago that primates emerged, and humans only about 0.4 of a million years ago. So that's way out here, almost at zero. So even though life on this planet has been here for 3.5 billion years, intelligent life has been here just for a flash, a tiny little slice of that time. So the likelihood of life, something being alive, is much higher than the likelihood that that life is intelligent and could communicate with us.
Damn.
Yeah.
What if that means the likelihood of us seeing dinosaurs is higher than intelligent life?
Say that again.
But I wonder if that means the likelihood of us seeing dinosaurs again is higher than intelligent life.
Yeah, it could be, especially since we now have perhaps the capability of making dinosaur DNA and reconstituting a dinosaur.
And but we're not too far away from Jurassic Park.
You know, when you say intelligent life, do you specifically mean just more like apes or humans who are using tools and stuff? Because then you could also look at things that are intelligent, such as mycelium networks that replicate, you know, like a brain's neural network that go over hundreds of miles is one of them, right? So what could be intelligent, but we just don't understand, right?
Yeah, there could be forms of intelligence that we don't understand, but it doesn't matter for purposes of this discussion how you define it, because any form of intelligence is at the very end of this time scale.
Gotcha, I understand, gotcha.
Is this the topic that's going to be on the next quiz?
Yeah, well, there might be something. I don't expect you to memorize billions of years, but the concepts here, I do hope that you'll understand.
Thanks.
Okay, and so here's how I'd like to conclude this discussion. Is there life elsewhere beyond Earth? The probability that there is really life elsewhere is very high. And why do I say that? It's because the number of Goldilocks planets, that is, planets where everything is just right for life to happen, the number of those is very large. And that's because the number of planets is astronomical, just beyond imagination. The amount of mass in this universe and the amounts of that mass that are constituted by planets. So because the numbers are so extremely high, it's very likely that life exists elsewhere.
I have a feeling we're going to get an answer to that at some point in time.
Well, let's look at the next issue here. What's the likelihood that we'll ever detect life as humans? And my estimation is it's very low. And why do I say that? Well, I've already mentioned the two issues. One is distance. Interstellar distances are so great that it would take us, given the technology that we're aware of, it would take us way too long to ever get to some place that has life on it. So distance is one huge impediment.
Professor?
Yes?
You know, when you say, okay, at the time before taking this class, I really wasn't interested in the other planets, whatever. But then they had two real rich guys that could take a trip to another planet. So does that mean it's able to host humans, or they were just viewing the other planet? It was only two very rich guys. I forgot what their name were. That's why I said I wasn't really interested at the time. But I remember them having the opportunity to go and visit or go and see another planet.
Yeah, and
I remember seeing it on the news. I just can't remember I can't remember, but I remember seeing that on the news where at least two people were going to go and visit with the astronaut, whatever, to see another planet.
Yeah.
Oh, so I'm not the only one to remember that. Okay, good. I'm not stupid. Crazy.
The issue you're talking about is impacted by distances. The distances are so great, and if you want to go to another planet, you want to go to one that had that you knew from observation had Goldilocks conditions on it. And to go to a planet like that would take an inordinate amount of time because the distance is so great. But the other factor I want to throw in here is what life forms are we expecting to find on any given planet? So we can just go back to here. So say life formed on another planet 3.5 billion years ago. And for most of that 300 billion years, life forms can't communicate with us. So the only way we can get evidence that they exist is to go find them, which brings us back to the problem of distance. We don't have enough time to travel the distances that would be needed to find primitive or unintelligible life forms. And so to find a planet, or for a planet to exist that was trying to find us, then becomes exceedingly small. So life forms likely to be primitive at any given point in time.
As Steve Stinson, but my question is, but if this billions of years has occurred in our solar system, our galaxy, then wouldn't the same thing apply for the time expanse for all those other galaxies and solar systems?
Yes. Yeah, they're all under the same constraints.
So wouldn't it be, I know the odds are astronomical, but based on that one swatch of, dang, I forgot what it was called, but galaxies, for lack of a better term, there were enough opportunities within that. I know 186F is the one that we could see, but beyond that, aren't there others that exist? In other words, with similarities similar to that we wouldn't even know about because of the vast expanse of space and time?
Yes, and that's really one of the main points here, is that because the numbers of planets in the universe is so high, so many, it's very likely that many of them have Goldilocks conditions and that life has developed on those planets. But because the distances are so great, because the likelihood that in any case the form of life is not intelligent, it reduces the possibility of we humans on this planet ever detecting life. It makes it the probability much lower. And so, question, will we ever detect extraterrestrial life before we ourselves become extinct? We know that our time existing is limited. Our solar system has a lifespan. And we know that conditions on this planet will become incompatible for any form of life at some point. So will we ever find life elsewhere before we become extinct? And the one positive thing to think about here is that we already have space stations. So if we could make space stations which were were self-sustainable, so that they recycled everything, then in principle, we could establish a human colony on a space station and go on out into the universe and go on forever. All we would ever need is a source of energy, which would be whatever star we're closest to.
Professor?
Yeah.
No,
No, I'm Sean here. I know this is just a kind of a comment, but I noticed you said the real problem is distance. It also sounds like the real problem for us is our lifespan. Um, and I was just kind of as you were speaking, I was kind of thinking about this whole thing going, yeah, life in a biological sense, might be very difficult to find, but intelligence is still starting to come out in the form of electronic intelligence. Um, so I mean, maybe we as a species might not be able to, but if we created something else to go out that didn't have the same constraint of lifespan, then maybe we could reach out at some point.
Yeah. 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 It's fun to think about these things because you can sort of expand your mind and try to imagine what might be possible that, or try to imagine things that we can't even imagine. Put it that way.
Professor, I'm sorry, but like I know it's down further when you're talking, but you're saying that we couldn't maybe live on another planet because of distance, whatever. I mean, not that I believe in aliens, but if it's possible, if it was, because I don't really believe in them, that they could come on Earth and live like regular, because they're on our, our Earth, then wouldn't it be possible for us to go live on whatever planet they come from as well?
Yeah, that's a possibility. I'd say it's not impossible. But again, I point out the distance problem, which is not insoluble. It's possible that that perhaps we or someone could travel at speeds faster than the speed of light. Right now, our understanding is that nothing can go faster than the speed of light, but maybe that's not true.
Isn't that?
I will say this, Professor. As someone who constantly looks at how technology is evolving, I have a feeling that we're gonna get there at some point. Technology advances so fast in the last 10 years, so it's only a matter of time. I mean, some of the inventions are kind of crazy, so I wouldn't be surprised if they eventually find a way.
Not in our lifetime.
I also feel like there's other things, such as we only see like a certain frequency of light. We only hear a certain frequency of sound. Like there very well could be things on our planet that are just like invisible that we have no idea about. And like we would never know anything about it, because we have such a extremely small sensory understanding of what we think it is, because we're human beings, that like there's so many endless possibilities. Like I'll just go into a rabbit hole right now, so I'll stop. But
you make a very good point, and that is that most of the matter in the universe we can't even detect with our senses and with our instruments. So you probably all heard of dark matter and dark energy. Well, dark matter is just that. It's matter that we only know exists by indirect measurements. And so it could be that if 95% of the universe or more is matter that we can't even comprehend, then who knows what's going on in all that matter. And Anthony, I think this is the point you're making.
Yeah, that's my point. I could go on for days about this. I honestly like.
Yeah, well, it's a fascinating topic, and I'd like to go on for days, but we are out of time.
Yeah.
All right. Well, thank you, Professor, for all this.
Yeah, well, I hope it was of interest to you, and because it's not actually in the curriculum, but it's of interest to me, and I think it's exciting to think about.
Yeah, you have a good day.
Okay.
Nice day.
Take care, and we'll see you. This is Hump Day, by the way, so I'll see you on Monday.
Thank
you.
Oh, what?
Thank you. Bye.
Bye. Have a great day.
Did somebody have a question?
Yeah, I did. what, besides the quiz, what's due? Because I missed 5 minutes or so. Besides the quiz, what's due for next week?
I don't think I've assigned anything, so
Oh,
just, I have a long weekend.
Oh,
coming up,
okay.
So I might conjure up something for you.
Oh, no, that's okay.
So we don't have anything else except for,
All
Just
that I
study
have
for
planned
the
for you
quiz.
now is a quiz. I have a couple more topics to introduce next week, but
Okay, cool.
I haven't organized those yet.
Okay, great. All right, thank you.
so no more homework and nothing, just to study for the quiz?
Excuse me?
So no more homework or whatever that's due except for the quiz?
Just for the quiz, yeah. That's all I'm assigning right now, but as I told Steve, I might think of something in the meantime.
Oh, no. Okay, next Monday?
The quiz is Wednesday,
Oh, it's gonna be next week. Oh, we still got more
week
time.
from today.
Will we
Hey,
have more
Buddy Chat, thank
assignments
you.
to prepare us for the quiz?
What?
Will we have more assignments to prepare us for the quiz?
I don't have anything definite in mind. I might come up with something that I'll just
Like next week?
keep an eye on, or between now and Monday, if you see an assignment or a worksheet in your Canvas, then go for it. But right now, I don't have anything prepared.
Yeah, leave it at that. That's good right there. Yeah, yeah. That's good. Leave it there.
But to prepare for the quiz or any test, just keep in mind that I'll always be asking questions about things that I think are important. And how do you find out what those are? Well, you look at the slides and you look at the recordings of the class periods, and that will get you 90% of or more of what's going to be on any quiz or any test.
Okay, thank you.
Okay, see you Monday.
Okay, bye. Have a good
Bye.
weekend. Bye, guys.
Okay.
Yoga.